DNA holds our genomic information, and unfortunately, it's not inert and it's subject to damage. Because we want to be able to preserve our genomic information, cells have evolved this very intricate system called DNA damage response. And there are two main pathways that happen within the system: DNA repair and apoptosis. So if a cell has DNA damage, it can choose to use many different mechanisms to repair that damage. But in cases when there's so much damage, the cell might decide it's in their best interest to activate apoptosis, or in other words, its own cell death. My lab is really interested in trying to understand how pathways within the DNA damage response talk to each other. We have a specific interest in altered DNA structures. So DNA, primarily the structure is a duplex, meaning it is comprised of two different strands of DNA. But under some biological processes, cells can actually form altered structures that are different from this duplex. Our lab is focusing on triplex DNA, so that means that instead of having two strands of DNA, the cell then has three strands of DNA. And that three-stranded structure is actually recognized by the cell as DNA damage. And we are trying to understand what proteins involved in repair actually are also essential for determining that repair can't be efficient. We now need to incorporate and alert apoptosis that it needs to step in order to preserve genomic integrity. My lab is really using molecular biology and biochemistry techniques to try and understand repair proteins. We use microscopy to look at these proteins on a cellular level and to see where they go within the cell. We also use those microscopy techniques to be able to understand where the DNA damage is, how the damage is getting repaired. And we look at this in many different cancer cell lines, in many different cancer types. In particular, my lab is interested in breast cancer and ovarian cancer. In addition to looking at things on a microscopic level, we're also interested in looking at things from a genomic level. So we do bulk RNA sequencing and looking at different genomic techniques so that we can actually see where in the genome across the whole entire cell is this damage happening. There are several different types of cancers, and one type of cancer are cancers that have gene amplification. And so what that means is that the cell has determined that in order for it to grow very aggressively, it will have multiple copies of a gene, and that gene gives it the growth advantage. Traditionally, drug development strategies have been to target those overexpressed protein products that happen as a result of the amplified genes. This has been very powerful, but it has been a limiting factor for many other types of cancers where small molecule targeting of those proteins is not effective and it's not doable. And so what my lab has been doing is to find ways to really target these cancers on a genomic level. We can design these synthetic oligonucleotide molecules that bind sequence specifically to the amplified gene. This creates a triplex structure. We know that the triplex structures, if you have enough of them formed, can cause the cell and force the cancer cell to activate its own death. We can use this system of manipulating DNA repair and apoptosis to cause cancer cells to die. I think it has far-reaching implications because there are over 450 genes that are amplified within cancer cells in more than over 14 subtypes of cancers. So this platform has really broad-reaching applications.